I'm Marguerite Matthews, a section chief in the NINDS's extramural division. I am thrilled to introduce today's WALS speaker, Dr. Moghaddam, who has made remarkable strides in revealing the mechanisms for adolescent onset of dopamine abnormalities and psychosis. Before I get started in my formal introduction, I have two very quick announcements. The first is the code for continuing medical education credits. The code is 57671. 57671. Also, for those watching remotely, you can submit questions to the speaker anytime during this lecture. Simply click on the button that says Send Live Feedback, which is below your videocast window. Type in your message and we'll relay it to the speaker at the end of the lecture. Now to our guest of honor. Dr. Moghaddam is the Ruth Matarazzo Professor in Behavioral Neuroscience at Oregon Health and Science University in Portland, Oregon. Her research employs a systems neuroscience approach to study dynamic brain mechanisms that maintain cognitive and emotional functions in key brain regions implicated in illnesses such as schizophrenia, ADHD, anxiety, and addictive disorders. Her primary focus has been on prefrontal cortex subregions and dopamine neurons in the midbrain. She has been an advocate for the development of relevant animal models in the pursuit of understanding the human brain. Exciting new research directions from her lab include characterization of neuronal systems from adolescence, the subject of today's talk, namely an exploration of the gene GRIN2A, which encodes the GLUN2A subunit of the NMDA receptor. Dr. Moghaddam has an undergraduate degree in chemistry from Avila College and a PhD in biochemistry from the University of Kansas. Rock, chalk, Jayhawk to all those who celebrate here and virtually. She completed her postdoctoral training in pharmacology at Yale University. She served in faculty positions at Yale and at the University of Pittsburgh for several years before moving on to OHSU in 2016 to become chair of the Department of Behavioral Neuroscience and now serves as the Ruth Matarazzo Professor since 2021. She has been continuously funded through her career with funding that has included a coveted NIH Merit Award and grants from the National Institute of Mental Health. In fact, the work she'll be discussing today is focused on a project she started as an assistant professor and has been funded by the NIMH that she still has today. So let that be some encouragement for those of you who are out there thinking about moving on to the faculty ranks. Dr. Moghaddam has been a supportive member of the neuroscience community. She sits on numerous committees and advisory panels, and she has been on executive boards of the Alliance for Diversity and Equity in Brain Science and the Schizophrenia International Research Society. She is also the American College of Neuropsychopharmacology's 2023 Dolores Shockley Diversity and Inclusion Advancement winner. On a personal note, I have come to know Dr. Moghaddam as Beta, a cherished mentor, friend, and confidant. Not only was she my PhD advisor at the University of Pittsburgh, but she also allowed me to talk her into moving to Portland, Oregon from Pittsburgh, where I was doing my postdoctoral fellowship in the Behavioral Neuroscience department. And before I actually moved here to NIH. So it is with great pleasure and honor that I introduce you to this brilliant scientist as her biggest fan, as she talks to you about the evolution of glutamate models of psychosis. Please welcome Dr. Moghaddam.
Thank you, Marguerite, for that wonderful introduction. Let's see. Sorry, we had a little bit of, no, we're good. We had a little bit of technical difficulty, but everything is working, no worries. And I taught undergraduates for years, so I can always do it. Chalk talk, no problem. So thank you for that wonderful, wonderful introduction. It's truly an honor to be here and have the chance to tell you a little bit about some of the ongoing work in my lab. I would like to start with some acknowledgments. First and foremost, I'd like to thank the American taxpayers for underwriting my research for over 30 years. Through both NIH fundings as well as DoD and NBA grants. I'm grateful for the generosity of individuals who donate to universities and private foundations. And last but not least, I am truly grateful to my trainees because they're the ones who are doing the exciting work. For today's talk, I will be primarily ending with the work of Michelle Keelholt, who is the latest PhD graduate from the lab. She just completed her dissertation a few months ago. So I thought today I would do something slightly different, which is that instead of telling you a specific science story, is to tell you the life story of an NIH grant, project MH48404. The 0 was added later as NIH started giving out more grants. So this beloved grant was my very first grant. It was, the title of it was Neurochemical Effects of Antipsychotic Drugs, and it was funded from 1992 to 1997. I was delighted when a few years ago this grant was renewed for years 29 till 34, and I'm currently at year 31 of the grant. So what I'm going to tell you, don't worry, I'm not going to tell you about 31 years of research, although I could. But I thought what I would do is to actually tell you about the bookends of this project, how it started and where it's going now. So let me start by telling you about the interest, why the interest in schizophrenia and antipsychotic drug research. Well, schizophrenia is a debilitating brain disorder. Some would argue that it is the most debilitating of all brain disorders, because its onset is early in adult life. So it poses a lifetime of anguish for individuals who have the illness and for their family. Obviously, because of this, it's also a very expensive disorder. It is also a deadly condition. It reduces life expectancy by at least 10 years. And it's also quite prevalent. Depending on who collects the data and where, it afflicts about 1 in 100 to 300 people. So obviously, that's one of the reasons that I've been interested in schizophrenia, but I was trained as a chemist. And so kind of shifting directions to neuroscience and schizophrenia was also very much influenced by my mentors. In particular, my PhD advisor, Ralph Adams, the University of Kansas. He was a truly pioneer electroanalytical chemist who invented many techniques, including in vivo voltammetry, as well as HPLC with electrochemical detection, which revolutionized the way we do postmortem analysis of brain tissue for monoamines. But he became very interested toward the end of his career in the biochemistry of schizophrenia, and that's when I was actually in the lab. So he was very influential in influencing my thinking about changing the direction of my research to schizophrenia. And another influential person was Pat Goldman when I was at Yale as a postdoc. She was not a schizophrenia researcher, but she also started being interested in it and started looking at it differently in a way because she started looking at it as a cortical illness, which again very much influenced my thinking. So for those of you not in the field, let me just give you a very brief history of schizophrenia. The first time it was introduced in the clinical literature as a distinct clinical entity was in the late 1800s when Emil Kraepelin coined it as dementia praecox. And then Eugene Bleuler, the Swiss psychiatrist, was the first to coin the term schizophrenia. Treatments for schizophrenia were suboptimal, if not cruel. They involved things like various shock therapies, the early versions of electroconvulsive therapy, and then in the 40s and 50s, what became quite prevalent was frontal lobotomies. So when antipsychotic drugs were discovered in the 50s, it was truly revolutionary. It was obviously revolutionary because these drugs, which we later found out or block dopamine receptors, obviously these drugs could have a substantial impact on the lives of patients because they were able to relieve some of the symptoms in a subgroup of them. But they also started really very much informing brain sciences and neurosciences because we appreciated that by actually targeting different brain receptors, we may be able to treat symptoms of a very serious mental illness. It was later obviously led to the dopamine hypothesis of schizophrenia, which remains quite prevalent and influenced much of the research related to the topic. So the idea was that, okay, if we understand how antipsychotic drugs work, then we can improve the lives of patients and design better drugs. And we were sort of on the roll because a few years later, the atypical antipsychotic drugs came on the market. It was discovered that clozapine, which we had known about that since the 60s, but had not been used a fair amount because it was associated with side effects like psychosis, that it actually potentially had better clinical efficacy than typical antipsychotics. So that led pharmaceutical companies to rushing to designing clozapine-like drugs, and that then led to the discovery of many drugs that are in use today that are all considered to be atypical antipsychotics. And these drugs, to this day, remain the main mode of treatment. So after all these years, we are pretty much are where we are. And in a way, we haven't made a lot of progress because we know that clinical efficacy of these drugs is quite limited in that they only treat the psychosis symptoms in a subgroup of patients, and also they have profound side effects. So 1990s, but there was a lot of excitement in the topic, is when I got in the field. And the first grant then was submitted then. And the idea that I had was actually at the time considered quite novel. Now you think, of course, glutamate is involved. But then it was actually considered sort of going against the grain, especially since I was at Yale, the mecca of dopamine. Everything was very dopamine-centric, that I started focusing on glutamate, synapse, and glutamate regulation of dopamine. Now, why focus on the glutamate synapse, and why did the study section actually decide to fund this when it was not the norm to study glutamate? Well, the norm was very much focused on dopamine neurons. And what I'm showing you here is for those of you who don't know about dopamine, which is to this day remains quite popular. Dopamine, even though it's very much loved and liked by many of us, it only comprises 0.001% of the neurons in the brain, a small subgroup of cells in the midbrain areas of ventral tegmental area and substantia nigra, with mostly unmyelinated axons that are sent to the prefrontal cortex and some basal ganglia regions. So the idea, the original logic I had was that something needs to be telling these dopamine neurons what to do. And being very much influenced by folks like Pat Goldman, of course, the boss of the brain was prefrontal cortex, and all projections from the prefrontal cortex were glutamatergic. So that's one of the reasons that I became very interested in studying glutamatergic regulation of dopamine. But at the same time, a few other pieces of data started coming out that very much supported this approach. An important one was the discovery that the psychotomimetic drug phencyclidine, and later MK801 and ketamine, block the NMDA subtype of the glutamate receptor. Glutamate neurotransmission is highly excitatory. It works to multiple receptors. One of the most prevalent ones is the NMDA receptor. Now, we had known about PCP since the late 40s when it went into the clinical trials as an anesthetic. Individuals woke up totally psychotic, so we had known that it's a psychotogenic or pro-psychotic drug. And we also knew that if patients with schizophrenia take it, the symptoms get much worse. So the discovery that the way it could be working and causing psychosis is through blocking the NMDA receptor was quite exciting, at least for those of us who were really interested in the glutamate neurotransmission then. There was a good number of postmortem studies were also trickling at the time, quite crude by today's standard, but they were also pointing to some glutamatergic abnormalities and not dopamine abnormalities. And last but not least, data was emerging from here, intramural program, Building 10, led by Karen Berman and Danny Weinberger, showing that there was disrupted cortical processing in schizophrenia. So I had my model, which was this drug that we sort of at the time thought, okay, this is how it's produced in psychosis. And then I had a circuitry to study. So then it made perfect logical sense, and the study section bought it, that it would be important to look and see how NMDA antagonists modulate dopamine neurotransmission and how do they disrupt cortical processing. Now, it sort of sounds boring, and I expected fundamental but boring results. Why? Because NMDA receptors are mediating excitation. So if you block them, you should see an inhibition. And that's, in fact, what everybody was seeing in the anesthetized animals and the slice work. The one thing that was different about my approach was that I decided to go to behaving animals. And this is a old slide because it's really an old slide. So anything that's super old from the first 5 or 10 years of the grant, I'll shift to the blue background because that was the fashion with PowerPoint in those days. So at the time, techniques weren't as fabulous as they are now, but regardless, I knew that I want to understand what happens to cortical processing in behaving rodents, in this case rats. So what I did was I essentially decided to look at the effect of PCP, ketamine, or MK1. These are all drugs that are NMDA antagonists. They're psychotogenic on various measures of in vivo measures as animals are behaving. And these involve measuring release of dopamine or glutamate or unit activity. And the idea was that they're going to cause an inhibition, and I wanted to essentially hoping to correlate the degree of inhibition or regional inhibition to various behavioral effects. But what I saw, which was one of the hugely surprising effects, was that there was no inhibition. There was profound excitation. Didn't matter what drug we used, which of the drugs we used, and what method we used, we saw an excess release of glutamate, excess release of dopamine, and also a good number of cortical cells fired faster. And I have just one example from those days, and it was a study that, we had a whole bunch of papers that actually looked at different drugs and different ways of looking at it, but the bottom line is that typical results was this. So what I'm showing you here, this is again old data, recording from a behaving rat, being given MK801. This is each line is one neuron. This is a heat plot, so as neurons fire faster, the colors get warmer. So as you can see, there's some cells that don't do anything, some that get inhibited, but the majority of them are getting excited. Okay. So this was exciting because it was entirely different than what we expected. We spent a fair amount of time working out the mechanism. A good number of papers came out. The mechanism that we came up with was that by disinhibition, there's excess release of glutamate that then excites the surrounding cells. It actually led to the disinhibition hypothesis, or supported the disinhibition hypothesis of schizophrenia. And to this day, these papers are used to understand potentially the effects of ketamine in depression. But for me, even though we were kind of trying to figure out the mechanism, the main question was, okay, it's exciting that we have this finding, but do these data have implication for therapy for schizophrenia? Because that's why I got into this area to begin with. So then the question that I asked based on the data we had was pretty straightforward, which was, by pharmacologically reducing this elevated glutamate transmission, what is the effect? Can we reduce the behavioral excitation, and then all these other excitation that we were saying? And if we can we actually then see if these drugs have clinical effects? Now, this being the 90s, it's also the heyday of glutamate pharmacology. About 10 years earlier, a number of glutamate receptors, including metabotropic glutamate receptors, had been cloned. Pharmaceutical companies were quite excited. Almost all the big ones had programs that were designing drugs because they assumed they will have, since we had started to learn a lot about glutamate receptor structures, that we could use them to treat Alzheimer's, stroke, et cetera. So we looked at a number of targets, but the target that we focused on, and this is an old diagram from actually 2003, but still is current because a lot of the colors you see, this is a diagram of a glutamate synapse showing some key proteins which we have direct or indirect ligands for. But the target we focused on, I should say we also studied, we looked at a number of targets, but the one that had the most interesting result was this glutamate M23 receptor. So the way these receptors work, they do a large number of things, but they're also localized presynaptically. And what we found was that when excess glutamate is released, this glutamate can then go and act on these presynaptic receptors, and then that presynaptic activation inhibits release. So a classic autoregulation of release. So the nice thing about looking at this sort of modulation is that you're not directly blocking or activating anything postsynaptically. You're just modulating some baseline release. So the drug worked beautifully. It really worked well in looking at behavior, looking at glutamate release, postsynaptic effects. And what was wonderful is that from 1998, when we had the very first paper, it took only 10 years for it to go to phase 2 clinical trials. I should emphasize that the first paper that we published, my co-author, Barbara Adams, was actually a technician in the lab. And because I thought this was a super risky project, it wasn't going to work, I did not give it to a postdoc. As an assistant professor, I did most of the work, and Barb essentially helped me. So it went to clinical trials after some open trials looked pretty good. So the phase 2 trials, the data was really exciting because the drug was compared to olanzapine, and after 4 weeks, the clinical efficacy was every bit as good as olanzapine, with the added bonus that it didn't have the side effects that atypical antipsychotic drugs like olanzapine had. So this was really exciting. I was happy. And what was really cool was that in the span of really 10 years or so, we went from looking at very, very fundamental basic studies to the first laboratory-based discovery of a truly novel drug, because it was not accidental. It was based on a model. Everything was clearly thought out. And it was also really nice and easy collaboration between my little lab as a little assistant professor and folks in academia, in industry, which at the time was much easier to collaborate with them, exchange drugs, and have them reproduce some of the findings that you had in the lab. Okay, so this was exciting. But what happened next was that larger phase 3 trial failed, and they failed badly. So Eli Lilly pulled the drug. All the other companies that had similar drugs also just pulled it out. So that starts one of the many OA phases of my research, where something that you think is super exciting doesn't work. So then the idea was, what next? Thankfully, I had tenure by then. And I don't just wish this upon someone who doesn't have tenure, but I had just gotten tenure. Regardless, it was disappointing. So the first thing I did, of course, was I declared a moratorium in the lab on animal models of schizophrenia. In fact, animal models of the disease were not mentioned in the lab, were not even mentioned in any grant that I submitted after that. And then the question is, okay, what next? Well, I decided that I really need to understand what is going on in the brain. It was disappointing because I had used what I thought was a clinically relevant model. I used the best techniques that were available to me. We had very mechanistic ideas, but everything failed. So it's like, at this point, we need to stop thinking about modeling. We need to go back and learn more about the brain. So one of the things that I actually started thinking a lot about was the temporal emergence of symptoms presentation in schizophrenia. And to explain that to you, I'm using a nice diagram from a review by Addington and Hyson, which plots stages of development, starting with gestation, puberty, adolescence, and young adulthood, and clinical signs and symptoms. So the progression is quite unique in that there are hardly any symptoms or measurable symptoms early on. Then early adolescence, the emergence of cognitive symptoms, some emotional, affective, and even motor symptoms. And then psychosis, which defines schizophrenia, does not typically happen until young adulthood. And this is the stage where antipsychotic drugs are introduced. So my thinking was that, okay, this is the stage I was studying, supposedly active psychosis, okay? But then, can we intervene to prevent illness progression here? And this is the direction that the field was also going. And conceptually, not that we could actually figure that out in my lifetime, is glutamate neurotransmission a pathogenic driver at this stage to then push this transition to full-blown psychosis? So one of the next ideas was that we need to understand a lot more about what's going on here. This is when I appreciated that the basic neuroscience of the adolescent brain was just pretty abysmal. We knew very little mechanistically about what happens to cortical processing, dopamine neuron firing, et cetera. Nearly all of our models, all of our studies at the animal level were all done in adult animals or really early in the stages of the development. So then we started looking at adolescent neuroscience of adolescence, and this is when Marguerite actually was in the lab, and over half of the lab was actually very much focused on the frustrating aspect of changing everything, all the methodologies that we have so that we can be looking at systems neuroscience of adolescent vulnerabilities. I think we had a number of outstanding papers that came out, and I'd like to think that they're contributing and continue to contribute a fair amount to our understanding of how cortical and dopamine systems are developing during this stage. But at the same time, of course, there was a moratorium about animal modeling, but it didn't mean that I wasn't thinking about it. I was thinking about it all the time. But I felt like I'm going to get back to it if I can do two things. Because like the whole idea for me, and you don't have to agree with this, but that's like for me, if a model, the point of a model in a basic science laboratory should not be to create an entire disease, but to test very specific hypotheses that are relevant to the disease experimentally. Also, in terms of designing the model, we need to use etiology, pathophysiology, symptoms framework to guide experimental modeling. Okay. So I'm happy to announce that the moratorium is lifted. So I'm here to actually tell you a little bit about this aspect of modeling that's going on in our lab, and primarily because we can use the etiology, pathophysiology, symptoms framework. Now, you're probably thinking, of course, that's how you need to be doing everything. But when it comes to understanding many of psychiatric illnesses, at least the research related to them, we're not doing that primarily because we don't know a whole lot about etiology, but we tend to very much fixate on specific symptoms and pathophysiology caused by either drugs or other manipulations. I was doing that with the whole PCP and ketamine model. I think the field of depression, a lot of our effort is spent on understanding how antidepressants work. Or with drug abuse, we spend a fair amount of time to understand how drugs abuse work versus what's about the etiology that actually promotes that drug seeking. But I think with schizophrenia, we are beginning to actually have the tools to start with etiology. So most of you may know that schizophrenia has a substantial genetic component. There's a high heritability rate that's estimated could be as high as 80%, depending on the populations that are studied. And these are attributed to common risk alleles. So they could be common, so there's just a simple association. But there's also many rare recurrent copy variants that have been recently, not the last 10, 15 years, have been verified that are very strongly associated with schizophrenia. In the last few years, thanks to a fair amount of collaboration and funding from institutions, including NIH and others, the power of these genetic findings is increasing. And what has been really exciting was that these two papers, at least exciting for me, that looked at common and rare variants, and they showed convergence, and I need to really emphasize convergence, of a few genes that both the common alleles and also the rare variants were associated with schizophrenia. And one of those genes is the GRIN2A gene, which is the subject of the talk today. So let me just show you another graph to emphasize the importance of how this works. So this is actually from a review by Hall and Bray, and they published this review before these last two Nature papers came out, which actually confirmed these findings because they had the higher N, essentially. So here they're plotting odds ratio for schizophrenia from different genes versus risk allele population frequency. So GRIN2A, the common regulatory variant, influences disease risk modestly. Okay? But then GRIN2A, the more damaging protein truncating variants and missense variants increase the risk substantially. And this is beautiful because it shows dose dependency, and these are loss of function genes. So dose dependently, loss of function of this gene could lead to schizophrenia. So this was exciting, and of course for us it was exciting because of what GRIN2A gene does, because we are back to our beloved NMDA receptor. The GRIN2A encodes the GLUN2A, depending on your age, you've seen this in the literature as NR2A, subunit of the NMDA receptor. Now NMDA receptors are these heteromers that have an obligatory NR1, so they all have to have this NR1 subunit. But they also have several NR2A subunits, and what they do is that they, depending on the type of NR2 subunit that they have, the biophysics of the NMDA receptor channel is modified. So the shifting the NR2 subunit can modulate the receptor slightly. The receptor still works, the channel is still open, but the modulation is disrupted. So this is really exciting because it was like, oh, all those years of thinking about an NMDA receptor wasn't a waste. But there's another really cool thing about this protein, or this, the gene and the product protein of GLUN2A, is that its expression is developmentally regulated. And we've actually known that for a while. Now, what do I mean by developmentally regulated? Since the late 90s, we have known this, that this GLUN2A, it's not expressed at birth, but it starts being expressed after birth, and there's a very steep rise, and it reaches adult levels in late juvenile, early adolescence, mostly late juvenile. And I'm showing the data from rats here, but this has been shown in other mammals, including post-mortem studies in humans also confirm that there's hardly any expression at birth, but there's a steep rise at birth, and it reaches adult levels in late childhood, early adolescence. So this is really exciting in the context of schizophrenia. Why? Because let's look at the temporal emergence of symptoms. If a primary cause is GLUN2A loss of function, so you're making less GLUN2A protein, of course you wouldn't have any symptoms at birth or early childhood because your brain normally, naturally is not making any. But as you age, then the normal levels start being produced. So if you're making less of it, then symptoms start being emerged. So that was quite exciting, and I should emphasize that there were several, after this finding came out, there were also several GLUN2A brain-wide developmental knockout mouse models, and these animals have pretty profound cortical deficits and movement abnormalities. But a real interesting question when this data began to emerge for us was, okay, so we can maybe explain what's going on here as you go into a expression increases. If there's a loss of function, you see these symptoms. But why are these dopaminergic abnormalities and psychosis happening later? And why should GRIN2A do anything to dopamine, or does it do anything to dopamine? So this is when we went back to, this is when the moratorium was lifted because then we could actually go back and think about very specific experimental questions. So the experimental question we had now was, what is a phenotype of a loss of function specifically in dopamine neurons? Of course, if you don't have it anywhere in the brain is all messed up. But what happens to the dopamine neurons per se if you have less of a GRIN2A? And given the wonderful methodologies that we have these days, then the model, and this is when Michelle Kilhol's dissertation work stepped in, we could actually have a very specific model to test this question, which is to have a GRIN2A knockout specifically in dopamine neurons right during adolescence. So the way that we have been doing the studies, and for the remainder of the talk, I'm going to briefly just mention some of the findings about with doing this highly specific manipulation in dopamine neurons adolescents. So we use a Cree-driven, CRISPR-generated virus to manipulate GLUN2A, and we're using the studies that I'm going to tell you about. We're using Th-Cree animals, meaning that if we actually infuse the virus in where dopamine cells are, they express TH, so the the virus is selectively taken up by dopamine cells. And this is how you can actually have cell-specific uptake of this gene. We infuse it right after weaning, so early childhood, you could argue, and then phenotype the animals during adolescence and early adulthood. So, obviously, Michelle did a lot of work for this, and a good 2 years was spent characterizing this virus. I'm not going to tell you about that for the interest of time, but if you're interested, we have a preprint in bioarchives that describes the details of the virus classification. But suffice it to say, the virus worked beautifully, and there was a reduced function of GLUN2A or GLUN2A in dopamine cells. Okay. So now keep in mind that this subunit of the NMDA receptor is just shifting the modulation of the NMDA receptors. You still have functional NMDA receptors on dopamine cells. It's just that they should be a little bit off. Okay. So what are the behavioral phenotypes? Well, we started by just looking at some basic characterization. The animals essentially looked fine. There were no gross abnormalities. So if you put them in an open field, look at their spontaneous activity in different maze settings to look at if they explore normally, they do. They move normally. So in a way, this was good because we weren't like deleting anything. We weren't disrupting the striatum or cortex, et cetera, in a gross way. Another thing that was important is that they were also okay eating and getting reward. So an example I have here for you is just putting the animals in a Skinner box. When the cue comes on, the animal has to execute an action in order to get a reward. As they figure out that this is the rule, the time spent between cue and action execution goes down because they know which end is up with the task. So we found that they both learned the same way, and so very simple reward-guided learning is not disrupted. So in a way, some of the basic fundamental aspects of the brain were working. So this was good, but then we wanted to see if maybe this just doesn't do anything, so which would disprove the hypothesis that this is actually a good model for anything, or that dopamine is involved in any aspect of anything related to schizophrenia. So that brings us to the fundamental question that, okay, what are the valid behavioral phenotypes for psychosis? Now, we are using rats. Rats are super smart animals, much smarter than mice. We can get them do a lot of really interesting things. They can also, they're happy to stay in an open box for hours. And so, but the bottom, psychosis, many would argue that it's a uniquely human condition. And a rat is not a person. So then the question was, okay, so how are we going to actually be phenotyping our animals in a way that's relevant to psychosis? Okay. So, and this is where I would like to really emphasize the importance of behavioral neuroscience training and also thinking thoughtfully about behavioral neuroscience, as opposed to just looking at very silly behaviors in mice and making a big deal out of it. But so we actually went and looked at, okay, what are some of the components or constructs that are relevant to positive symptoms or psychosis in schizophrenia? Well, there are delusions and hallucinations, and the constructs that are relevant to delusions and hallucinations can include, are not limited to, but can include faulty inference, aberrant salient attribution, and impairability to use feedback or representation to guide behavior. So these are constructs we could actually test in a smart rat. Thought disorder, I'm not even going to begin to start testing thought disorder in the rats, so we know our limitations. But then the third construct relevant to psychosis is abnormalities in perceptual organization. And again, constructs associated with perceptual organization can include a decreased influence of context, difficulty optimizing effort allocation, and aberrant salience. So what I'm going to do for the limited time that I have is just touch upon a few behaviors that were positive or negative data related to some of these, and also end with just measuring dopamine released directly in these animals that we had knocked down the GRIN2A during adolescence in dopamine cells. Okay. So the first task I'm going to tell you is a well-known task. It's called progressive ratio task. It's very popular by the drug abuse colleagues. It's been around for a long time. It's pretty straightforward in that you actually put an animal, again, in a Skinner box where they have to respond to get a reward, except that the number of responses gradually goes up. It actually goes up exponentially. So they have to keep responding more and more and more and more in order to get the same reward. It's a very common task in the drug abuse field because it's a measure of how rewarding something is. Animals are willing to work harder, make more responses for cocaine, for example, versus food. So one thing we found was that our knockout animals, the break point, which is how many responses are animals willing to take to get the reward, was the same. So willing to work as hard to get a sugar reward. So this is essentially the data with 4 test sessions. But if you start looking at more aspects of this task, one of the interesting things that we found was looking at the so-called post-reinforcement pause. And I'm showing this phenomena in control animals. Now, what is post-reinforcement pause? As animals go and respond a few times and they get a reward, they know, they have learned the task. They know that they have to go back and respond a lot more next time in order to get it. So once you get toward the middle of the session, just put yourself in that position that, okay, you got in the sandwich. Do I need to go and work a lot harder to get another bite of that sandwich? There's a point where you start pausing and thinking, is this worth it or not? And this significantly goes up. This post-reinforcement pause significantly goes up in control animals. And one of the things we find is that in our lesion animals, this significantly is different, and that these animals, it just doesn't faze them that now they have to go back and respond 80 times. They just go back and forth, back and forth, back and forth, and they respond. So this is interesting because it could imply difficulty, obviously, optimizing effort allocation, because are you not sensing that maybe this is not worth my effort? Decreased influence of context, that you're not appreciating that, okay, it's later in the task, I need to work harder, should I actually do it? And then a reduced ability to use negative feedback, which is not, lever press 5 times, 50 times, and I'm not getting a reward. So it could imply any of those things. No. Then another task that I'm going to tell you about is Pavlovian now. This one, which is that there's no action involved. The animals just sit there, and a cue comes on, and there's an outcome associated with that cue. And the way the task we use, actually, Dr. Matthews here helped develop this task during her rotation. The animals are provided with 2 cues. And I will be calling them CS, conditioned stimulus. One is paired with a reward. The other one is paired with a mild shock. So in this case, sound is paired with reward, light is paired with shock. Of course, it's counterbalanced, so animals are getting one or the other, are getting both in different groups. And then after they learn it, we reverse contingencies. And the way to actually measure learning that they've appreciated is to actually look at, in this case, we're using an index that measures how quickly animals actually go to the feeder and get the food. So if you look at our data with our lesion animals, during the initial acquisition, there's not much of a difference. Here I'm showing you the stimulus associated with the reward that shifts to being associated with a punishment. But what happens is that the lesion then produces abnormal responding after contingency reversal. So even the learning that something's associated with a good or bad outcome is there, but there's an inability to actually shift contingencies. And what this does is, again, relevant to some of the subconstructs of the positive symptoms, is that it really implies that there's an impaired ability to use feedback or mental representation to guide behavior. And then what happens to phasic activity? So let me give you dopamine activity. Let me give you an example for that. In the same animals that are injecting the virus, we can also inject another virus, GRAB2A, so that later on we can use fiber photometry to measure dopamine as they do behavioral task. And this is important in the context of the clinical relevance of this manipulation, because we have a fair amount of dopamine-related data in patients with schizophrenia. Both PET studies have shown a lot of data, but there's also fMRI studies that have looked at reward processing. And one of the studies is actually looking at reward processing in nucleus accumbens. And what they find, and actually there's several studies that have done that, but the general conclusion is that reward activity itself is not affected. And we've seen very much quote-unquote normally related behavior in these animals. But there is a blunted ventral striatal activity when individuals are exposed to unexpected reward. So with that in mind, let me show you dopamine response in these animals during two events. So one is during novelty. The other one is during unexpected outcome. Now, novelty is interesting because when you hear something entirely novel, especially if it's auditory or a visual cue that you haven't heard or seen before, your dopamine cells fire. It's thought to be a very bottom-up, unfiltered phasic response. So we were interested to see if that effect, which is, again, very, you could say, automatic, is affected with this treatment. But then unexpected outcome at this session when contingencies shift is very much involves decision-making, remembering what happened in the session before, and actually changing your behavior and appreciating that contingencies have shifted. So you could argue that this is a far more top-down, complex behavior. So let's look at novelty. We see that novelty is not affected. So the data I'm showing you here is response to stimulus that is the two stimuli that are associated with either shock or reward. In session 1 and in trial 1 to 5, the very first time the animals are exposed to these stimuli, expected beautiful phasic response. And then as the session continues and these stimuli are no longer novel, it goes down. So clearly the stimulus bottom-up regulation of dopamine activity has not changed. Now let's look at the unexpected outcome. Here we looked at the dopamine activity in early and late sessions. So what happens in session 6? Animals for the first time are getting exposed to a different outcome associated with what was familiar. So the stimulus, they go on session 6, they go in the box, and let's say the light, they thought it's going to produce reward, but now it produces shock. So we looked, but then toward the end of session 6, then they have actually made the association. They realized that it has changed. So if you look at early session 6, this is when expected reward, the animals expected a reward, they received a shock. You see that there's a difference, that this going to a lesion animals are having a muted response. And the same with when they expect a shock, but they receive a reward. So there's clearly a difference. There is this muted response when a contingency is unexpected or novel. But as we run the animals toward the end of session 6, that difference disappears. So once they learn, then that disappears. And I'm showing this example because it's for me, it's really interesting because it very much agrees with data that we have seen in patients, which is that there's a blunted ventral striatal activity very selectively during unexpected reward. Yeah. So I'm going to end by then emphasizing that we are at a stage in schizophrenia research that we have actually enough knowledge to start with targeting some relevant etiology and then understand what it does. I hope today I convinced you that we can actually do this with the green 2A, that we can very specifically go and knock it down in adolescent dopamine neurons and have a phenotype. And I appreciate that the behaviors are subtle, but in a way that's good. But the phenotypes are quite specific, and including dopamine release, and they are relevant to some aspects of the positive symptoms. And I think this general direction for the future is exciting because it represents a clinically valid etiology model that could specifically, remember these are adolescents, could specifically target the emergence of psychosis in vulnerable adolescents. So this is where I left you off, and there was a failed trial. And now, and a lot of really, really good papers did happen here. They've been cited a lot, primarily because they were focusing on adolescent neuroscience. But I think now we are beginning to develop ideologically relevant animal models for latent expression of psychosis. Okay, so a little bit of my ideas about moving forward. I think this way of approach has implications for precision psychiatry as genotyping becomes cheaper and cheaper, and artificial intelligence is entering the way we are generating dealing with data analysis. I think it also has important relevance for potential of using animal models, not to just come up with a better antipsychotic drug, but potentially develop and screen course-altering treatments that could prevent onset of psychosis. Now, I appreciate that pharma has very much pulled out of, at least big pharma, developing glutamatergic drugs, these sorts of drugs. But there's a fair number of targets that are there because they did remain up to a few years ago interested in developing glutamatergic drugs for models related to Alzheimer's and MS. I'm just showing you a few papers of these are Genentech drugs that are targeting the product of GLUN2A receptor. Now, none of these targets are ready for prime time. They're either are associated with toxicity or have very poor absorption. But regardless, they are there, and hopefully we can go back to them. And then last but not least, remember the sob story of this failed clinical trial? Well, because the phase 3 was a very large trial, there were several thousand subjects there. There were a number of post hoc analyses that were performed. And one of the most interesting ones was that the drug actually did work early in disease, and that they showed significant improvement. So I just grabbed one figure from that analysis. The paper was published, this data was published in Biological Psychiatry a few years later. So if I bring your attention here to comparing early in disease, given this mGlu2/3 agonist, versus late in disease, look at the blue line, which is risperidone, which is the comparator typical antipsychotic, and then the red line, which is the LY drug. They're very similar. It didn't work. The red and blue separate. It didn't work in older patients, but it seemed to have worked in younger patients. So with that in mind, hopefully moving forward with a better understanding of the etiology of the illness, hopefully we can also rethink many of the previous failed clinical trials. And this one was not the only one. There were many more. So I'd like to end, again, acknowledging my wonderful lab. These are the name of a few people who have been actually supported by the grant. The current, some current pictures of a very good-looking lab. And for this project that I told you about, we're very much indebted to amazing collaborators. Larry Zweifel made us all the viruses that we have used. Angela Langdon helped us a lot with the behavioral modeling that I didn't have a chance to talk. And then also John Williams was very helpful in helping us to establish the feasibility of using this virus. So I thank you for listening, and if there's any time, I'm happy to answer any questions.
Yes. Plenty of time. We welcome anyone to use the mics. There are a couple. Yeah, please. Please. Yeah.
Thank you for a wonderful talk. Given the importance of dopamine in mood control and reward circuitry, is it possible to treat schizophrenia without these adverse effects that affect the dopamine pathway?
That's a great question. Obviously not, because we have been giving them dopamine antagonists, and although they improve symptoms that are relevant to psychosis, some may argue that they worsen affective and cognitive symptoms, which is more the reason to actually go beyond the dopamine system. And if for a subgroup of patients, a primary reason that is causing the symptoms is GLUN2A loss of function, then we could, for that subgroup, we could be targeting GLUN2A specifically.
Dr. Raza?
Hi, thanks very much for a great talk. And you've spoken about the importance of studying glutaminergic and dopaminergic systems in the context of the kind of adolescent unfolding and emergence of psychosis. And your work very kind of precisely targets what might be going on in that window. Mm-hmm. How do you think about their role in the setting up of the system neurodevelopmentally? Mm-hmm. And do you think that's a sort of necessary primer against which to think about the adolescent effects?
Yeah, I think it is actually. I didn't have the chance to show you the data that actually, and it's in the bioarchive preprint. GRIN2A expression is very different in the midbrain than it is in cortex. So in cortex and striatum, we've known since the 90s that it goes up and stays level. In dopamine neurons, in adolescence, there's a big dip normally. So we think that there's actually, in the midbrain areas, at least in dopamine neurons, there is this quote-unquote normal shift in GRIN2A expression so if you have then a loss of function of GRIN2A, then that period, late adolescence, is when your dopamine neurons are most vulnerable, whereas earlier in development, like in childhood, is when your cortical neurons are more vulnerable. So the answer is yes, and we have some data to show. And if anybody is interested in collaborating and looking at it in a more reductionist way, I'm happy to do that.
Thank you for the talk. It was very fascinating. Oh, sorry. Thank you for your talk. It's very fascinating. So I'm curious about the role of other neuron populations, like serotonergic neurons and therapies that are being developed for that, particularly with psilocybin, and I saw that you do some work with that. What do you think, if any, overlap between the therapeutic benefit of psilocybin and schizophrenia, and many overlapping pathways?
Well, psilocybin could certainly make him a lot worse. So it's a pretty pro-psychotic drug, and there are several papers coming out that there's actually an uptake of psychotic symptoms in young individuals who are taking psilocybin. Obviously, it's not related to this talk, and I have struggled to get any funding, government funding for psilocybin, but thankfully there's private funding for it. We have some models for the pro-psychotic effects of psilocybin, but that doesn't answer your question about other neuronal pathways. We have some data actually targeting GABA neurons in the VTA, because we also have GAD-CRE lines, and they do have a slightly different profile. So I think that, in my opinion, the beauty of this sort of approach is that you can mechanistically separate what happens if you have a loss of function of specific genes like GRIN2A, what happens to dopamine versus serotonin versus GABA neurons in different brain regions. And I think I'm not sure if it would have relevance for therapy, but it would clearly have relevance for understanding of the progression of the pathophysiology of the illness.
Thank you.
But the viruses are available. If anybody wants to look at them in different cell-specific way, that would be great. I've been afraid of serotonin for a while, so my lab is not going to go there. So
there's a lot of excitement about M1, M4 agonists right now in the schizophrenia field. What are the connections between that system and the glutamatergic system?
Yeah, so M1, M4, there's excitement because it was approved it is accidental discovery. It's a muscarinic receptor targeting drug, and it was accidental because I believe it looked well in patient. It went for trial in Alzheimer's patients, and then it improved their psychotic symptoms. So I wish the mGlu trials were done as that drug were, because if you look at the exclusion criterion for that clinical trial, it was pretty impressive what they were able to do. So I think if this drug was not, and I'm just second guessing, obviously, if the trials were not it was not primarily in institutionalized, very ill patients in Eastern Europe and Russia, if all the exclusion criteria that were applied to that drug potentially could have helped this one. In terms of the mechanistic connection, I don't know, but it opens a lot of really interesting ideas. And then it could also be lovely if we have multiple approaches that, depending on your genotype, then we actually can have more precise treatment for individual patients. So the long answer to your question is we don't know yet, but there's potentially a lot of ideas for further study.
Okay, Thanks.
Thanks for your wonderful talk. So I have two questions here. So one of them, coming from a background of ketamine model of psychosis from your early work, does that ketamine model of psychosis recruit dopaminergic abnormalities, which has been established in your group as well? So have you tried any kind of occlusion experiment in your current dopamine-specific NR2A model, trying to do a ketamine-induced psychosis and see whether the ketamine recruits the NMDA receptors present in the dopaminergic neurons?
You mean in animals that have had GLIN2A knockdown?
Right.
Yeah, no, we haven't. So we've actually looked at NMDA recruitment and blockade in the slice to just see how well it works. But we haven't looked at a non-competitive. So we haven't. I think the way, I can kind of see where you're going mechanistically with that. The way to do that initially would be to actually just do some patch studies to see if you're stimulating these neurons, whether if you can block NMDA function. My guess is that you can still do that because ketamine goes inside the NR1 subunits. NR2 function has very little to do, unless something has changed in the last few years. NR2 subunit doesn't have a lot to do with ketamine actually being able to get into an open channel. So my prediction would be no.
Okay. The second question that I have is related to the GABA model of NMDA receptor hypofunction. So GABA neurons, they also express NMDA receptors, and a lot of labs point to the fact that hyperdopaminergia in dopamine neurons could be resulted from hyperexcitation coming from cortical regions as a result of reduced inhibition.
Yeah.
So when things are so complex, how would you how to mechanistically understand the same NMDA hypofunction being beneficial in antidepressant?
Well, because it's more complex than just NMDA hypofunction, and I think it involves, in this case, a gene like GLIN2A, which is expressed differently in different subsets of neurons. So as we find that it's being expressed differently in cortex at a different time than it is in dopamine neurons. So I think I'm hoping that we could just go beyond hyperfunction in a general way and very much focus on these very specific pathways that are more cell-specific and involve different developmental periods.
Wonderful. Thank you very much.
Hi, Bob.
Hi. I have an extremely naive question. So is anything known about why GLIN2A, the expression of GLIN2A is temporally regulated?
So there's some molecular work done in not as much as we should now, but there is some work done about 10, 15 years ago suggesting that there's kind of this shift between 2B and 2A, that 2B, the same transductional mechanisms that reduce 2B production now then enhance 2A production. That hypothesis seems to not to hold in all brain regions. So the answer to your question is not exactly, but I think molecular biologists should have the tool to actually look at that. And especially, it would be lovely to know what is it different about a TH expressing, which is a GAD expressing neuron during development, that one should start expressing this in early childhood, the other one in late adolescence, for example.
Yeah. So. Okay, thank you.
Okay, well, first, let me repeat the CME code, 57671. 57671. That's how you said it, Margarita. And we have a reception. Please join us right outside, and let's thank the speaker.
Thank you.